The work starts from our previous discoveries of widespread and diffuse genomic heterogeneity in multiple myeloma, which is a compelling evidence highlighted by recent genomic studies and has a bunch of translational and clinical implications if we want to understand more about disease biology and provide clinicians with I would say a decision-making framework that involves the latest genomic findings and may allow delivering to each patient the best care. So, of course, we have known for decades that the major cytogenetic categories in multiple myeloma depend on either the presence of hyperdiploidy or IgH translocations. Nevertheless, recent genomic studies have expanded immensely, I would say, the list of driver events in multiple myeloma, and that is genomic abnormalities that may have a consequence and change the way the disease behaves. So, taking advantage of the COMPASS study, which is an invaluable resource of genome sequencing and transcriptome sequencing over thousands of cases, we have been able to dissect the impact of each of those dozens of genetic abnormalities on the transcriptome, with the idea that whatever impacts gene expression and therefore protein expression is something that has an actual biological meaning. So what we found is that cytogenetic events, and again, hyperdiploidy and the translocations, have the most impact on gene expression. Surprisingly, we found that gene mutations, which have been the focus of many discoveries in the past 10 years, do not have a great impact on the transcriptome. And there are many ways to interpret these results. It may be that these mutations are so widespread and somehow redundant that it's hard to dissect the specific consequence of one. Or it may be that actually these mutations arise in a genetic background that has been already predetermined by the previous genomic alterations, which are mainly of structural abnormalities of chromosomes. Nevertheless, we were able to add some information to the notion of what are the consequences of gene mutations, because we did find that, for example, for many genes, the consequence of the mutation depends very specifically on the amino acid residue that's hit by the mutation itself. And it will be very interesting in the future to understand why, for example, in multiple myeloma, KRAS mutations occur preferentially at one amino acid, and in colon cancer, KRAS mutations occur preferentially at another amino acid. It may be that there are functional differences. Furthermore, we have known now for quite some time that tumor suppressor genes in myeloma are actually inactivated in both the maternal and parental allele. So talking about mutations, it's only revealing part of the story, because very often when one allele is mutated, the other one is deleted. And we analyzed these instances of biallelic inactivation, and we found that there is a cumulative effect of gene inactivation when both alleles are inactivated. And of course, these are instances of biallelic inactivation are the ones with the most striking effect. Looking at genes that are deregulated upon biallelic inactivation of tumor suppressors, we ran upon the gene BCL2, and that is an anti-apoptotic protein which has recently become a major player in multiple myeloma because it is a biomarker of the drug venetoclax. So people with high BCL2 expression responded to venetoclax, but so far we don't measure BCL2 routinely in the clinic. So we need genomic biomarkers to predict who will respond. And among those, we know that translocation 1114 is the most widespread and used biomarker for venetoclax response. But we have found that another gene, particularly CYLD, is a gene that, when deleted, has the most striking upregulation of BCL2 in our cases. So when we went on to produce in silico screens of cell lines and measure the response to venetoclax based on the genomic status of cell lines, and particularly myeloma cell lines, and we actually could validate how deletion of CYLD is a potential novel candidate of venetoclax response. So altogether, our study started as a bit of a fishing net thrown at transcriptome complexity in multiple myeloma, but we feel that throwing such a large net has produced findings that are actually of high translational relevance and will be the starting point for future experiments.